Let's welcome our upcoming speaker, Dr. Abdulhadi Alqahtani, clinical trial expert at Saudi NIH, playing a key role in advancing clinical
Assalamu alaikum. First of all, I would like to thank the organizing committee for inviting me. It's my pleasure to be with you today. Today, our topic, we will talk about basic of clinical research, and we will focus exactly about study designing and methodology approach. We will highlight high-level information in this presentation, because I think there is a workshop they will discuss in deep. As we all know, the clinical research or the importance of the clinical research in helping us to understanding the disease and how the treatments work, as well as if there is any side effect from this treatment or not. We will talk here about clinical research. We usually do a clinical research to investigate how disease and medical condition develop, as well as how they can be treated and prevented. Also regarding the healthcare interventions, how its impact on the patient. By doing any research, we have a study design and methodological approach or methodology. It's the backbone of any research. It's help us and guide us to what to do and what's the data to be collected, as well as how we interpret this data. Given that we are here in pharmacy conference, we need to understand what's the importance of clinical research for the pharmacist. As we pharmacy, we interact usually in clinical research with the medications or the treatment, and we want to understand these treatments, and we need to optimize the patient used for these treatments. However, we are looking for the improvement of the patient impact. There's two main types in clinical research, which is observational and intervention types, or study design. As we know, the intervention or observational study design are just is the researcher just observe and collect the data without interfere the subject. However, the intervention are the researcher actively influence or introduce the interventions to the participants. When we talk about observational study design, we are talking about there's exposure and there's outcome. And the advantage of the observational study design is to measure the real world effectiveness. And by using the observational, we can test or examine multiple exposure. However, the data, it's not 100% accurate in the observational study design, and we cannot make sure this factor or this outcome it's really caused this effect. and there is a lot of confoundings. As we see here, there is a potential for measurement bias, there is a potential for selection bias, as well as there is a potential for confounders. There's the three main type of observational study design: cross-sectional studies, and case control, and cohort study. If we talk about cross-sectional, that means we are seeking to identify prevalence or frequency in the snapshot within specific time, within specific group. And the advantage for cross-sectional, it's low cost, fast results, and usually we use it to know or to understand the prevalence. For the disadvantage, we cannot, as we mentioned, we cannot evaluate the incidence or the efficacy or the risk. Also, we cannot identify the causality. For the case control, we have when we want to do a comparison between two groups, both, for example, both are the outcome, and we want to see what's the exposure. Usually we use this when we want to see the retrospective information. The advantage of this is study rarely outcome, or rare outcome, and multiple exposure can be tested at the same time. For the cohort study, we are dealing with the exposure. We have a group of patients, then we have the exposure factor, and we will follow them for the certain time. Usually it's a longer time to understand the outcome of this intervention or the factor. We talk now about intervention study design. When we talk about intervention study design, we are focusing, or we want to evaluate the safety and efficacy and effectiveness of a new treatment. This treatment can be a drug or medical device. Usually clinical trial classified 4 phases, as my colleague mentioned. We will talk briefly for each phases. In phase 1, which is the safety and dosage, we usually do phase 1 to evaluate the safety of a new treatment and pharmacokinetics and pharmacodynamics, and to see the toxicity from this new molecule. Study design, we use a small number of patients in phase 1, and those patients might be voluntarily healthy, and for some conditions, serious conditions, we include a patient. We use open label in phase 1. Might be single site or multiple sites. Also, we are focusing here in phase 1 to dose escalating. We have 3+3 study design rule, or dose escalating. There is the 2 types of these dose escalating. First, we have initial dose for 3 participants. Then we will escalate it. If the escalation doesn't result any severe DLT, then we will go forward to have different group or different pathway. First, we have if the DLT development 1 among 3, we will do escalation for the dose. Then there is a 3 type. expand the cohort or the patient participate from 3 to 6, then see if it's developed in one or less among these 6, so the dose will be the same. So the dose will be escalated. If it's more than 2, the dose, we will stop the escalation, then the last dose will be considered the maximum dose. In the another hand, from the beginning, if the DLT progresses in more than 2 among those 3, we will stop the escalating, then we will consider the last dose its maximum dose. And usually this for few months. For this phase 2, we are looking for efficacy and side effect. We already have initial profile about the safety. Then we are focused or looking for the efficacy to understand the optimal dose of this dosage, safe dosage. Then we will go in the study design. We are here including hundreds of patients. In phase 2, we start with the patient with a targeted disease. Then we have a randomized clinical trial, and we have placebo control, either it's inactive or with a control treatment. Here in phase 2, there is a single and double blind. As we know, the patient doesn't know about the medication, and the double blind, there is the patient and the study team doesn't know about the medications. Here, the important things in phase 2, we should have endpoint to say there is efficacy in this phase. Most likely, or sometimes we can have 0.5 or 10%, for example, decrease in hemoglobin A1C if we are targeting, for example, diabetic patients. This will make several months up to a year. In phase 3, what we call it confirmatory trial. That means we confirm about the safety, and there is a start point efficacy, or the dose of efficacy. It's already determined in phase 2. However, we start to exploring the effectiveness among larger group and with a different demographic and characteristic. We usually include 1,000 on phase 3. As we mentioned, randomized clinical trial is a study design, and there is the active or placebo control and treatment control. The double blind is the gold standard for the phase 3 trial. However, we have to determine the clinical outcome. Last phase, we are talking here about post-market efficacy and safety. We do usually post-market to maintain or to extend the efficacy and safety report for the treatment, and this phase usually done after the drug in the market to make sure the efficacy is still there and the safety within the population, among the whole population, it's there. Usually the study design of the post-market are observational and study registry, as well as long follow-up for those patients who is taking this medication, for example, to collect any new adverse event. And this it might be ongoing during the whole journey of the patient on the drug. By this, thank you so much. I conclude my presentation. As I mentioned, it's high level. So because I saw there is a workshop will include deep information. Thank you so much.